The epidermis is the outer of the two layers that make up the skin, the inner layer being the dermis. It provides a barrier to infection from environmental pathogens and regulates the amount of water released from the body into the atmosphere through transepidermal water loss. The outermost part of the epidermis is composed of a stratified layer of flattened cells. That ovally a basal layer composed of columnar cells arranged perpendicularly. In humans, it is thinnest on the eyelids at 0.05 mm and thickest on the palms and soles at 1.5 mm. It is ectodermal in origin. Structure. Cellular components. The epidermis has no blood supply and is nourished almost exclusively by diffused oxygen from the surrounding air. It is 95% keratinocytes but also contains melanocytes, Langerhans cells, Merkel cells, and inflammatory cells. Rete ridges are epidermal thickenings that extend downward between dermal papillae. Blood capillaries are found beneath the epidermis and are linked to an arteriole and a venule. Layers. The epidermis is composed of 4 or 5 layers depending on the region of skin being considered. Those layers in descending order are: cornified layer, composed of 10 to 30 layers of polyhedral enucleated corneocytes, with the palms and soles having the most layers. Corneocytes are surrounded by a protein envelope, filled with water-retaining keratin proteins, attached together through corneodesmosomes and surrounded in the extracellular space by stacked layers of lipids. Most of the barrier functions of the epidermis localize to this layer. Clear, translucent layer. The skin found in the palms and soles is known as thick skin, because it has 5 epidermal layers instead of 4. Granular layer. Keratinocytes lose the nuclei and the cytoplasm appears granular. Lipids, contained into those keratinocytes within lamellar bodies, are released into the extracellular space through exocytosis to form a lipid barrier. Those polar lipids are then converted into non-polar lipids and arranged parallel to the cell surface. For example, glycosphingolipids become ceramides and phospholipids become free fatty acids. Spinous layer. Keratinocytes become connected through desmosomes and start produce lamellar bodies from within the Golgi, enriched in polar lipids, glycosphingolipids, free sterols, phospholipids and catabolic enzymes. Langerhans cells. Immunologically active cells are located in the middle of this layer. Basal, germinal layer. Composed mainly of proliferating and non-proliferating keratinocytes, attached to the basement membrane by hemidesmosomes. Melanocytes are present, connected to numerous keratinocytes in this and other strata through dendrites. Merkel cells are also found in the stratum basale with large numbers in touch-sensitive sites such as the fingertips and lips. They are closely associated with cutaneous nerves and seem to be involved in light touch sensation. The term alpine layer is usually defined as both the stratum basale and stratum spinosum. The epidermis is separated from the dermis, its underlying tissue, by a basement membrane. Cellular kinetics. Cell division. The stratified squamous epithelium is maintained by cell division within the stratum basale. Differentiating cell delaminate from the basement membrane and are displaced outwards through the epidermal layers, undergoing multiple stages of differentiation until, in the stratum corneum, losing the nucleus and fusing to squamous sheets, which are eventually shed from the surface. Differentiated keratinocytes secrete keratin proteins which contribute to the formation of an extracellular matrix and is an integral part of the skin barrier function. In normal skin, the rate of keratinocyte production equals the rate of loss, taking about 2 weeks for a cell to journey from the stratum basale to the top of the stratum granulosum, and an additional 4 weeks to cross the stratum corneum. the entire epidermis is replaced by new cell growth over a period of about 48 days. Calcium concentration. Keratinocyte differentiation throughout the epidermis is in part mediated by calcium gradient, increasing from the stratum basale until the outer stratum granulosum, where it reaches its maximum, and decreasing in the stratum corneum. Calcium concentration in the stratum corneum is very low, in part because those relatively dry cells are not able to dissolve the ions. This calcium gradient parallels keratinocyte differentiation and as such is considered a key regulator in the formation of the epidermal layers. Elevation of extracellular calcium concentrations induces an increase in intracellular free calcium concentrations. Part of that intracellular increase comes from calcium release from intracellular stores and another part comes from transmembrane calcium influx through both calcium-sensitive chloride channels and voltage-independent cation channels permeable to calcium. Moreover, it has been suggested that an extracellular calcium-sensing receptor also contributes to the rise in intracellular calcium concentration. Development. Epidermal organogenesis. The formation of the epidermis begins in the cells covering the embryo after neurulation. The formation of the central nervous system. In most vertebrates, this original one-layered structure quickly transforms into a two-layered tissue, a temporary outer layer, the periderm, which is disposed once the inner basal layer, a stratum germinativum, has formed. This inner layer is a germinal epithelium that gives rise to all epidermal cells. It divides to form the outer spinous layer. The cells of these two layers, together called the Malpighian layer after Marcello Malpighi, divide to form the superficial granular layer of the epidermis. The cells in the stratum granulosum do not divide, but instead form skin cells called keratinocytes from the granules of keratin. These skin cells finally become the cornified layer, the outermost epidermal layer, where the cells become flattened sacs with the nuclei located at one end of the cell. After birth, these outermost cells are replaced by new cells from the stratum granulosum, and throughout life they are shed at a rate of 0.001 to 0.003 ounces of skin flakes every hour, or 0.024 to 0.072 ounces per day. Epidermal development is a product of several growth factors, two of which are: transforming growth factor alpha is an autocrine growth factor by which basal cells stimulate their own division. Keratinocyte growth factor is a paracrine growth factor produced by the underlying dermal fibroblasts, in which the proliferation of basal cells is regulated. Function. Barrier. The epidermis serves as a barrier to protect the body against microbial pathogens, oxidant stress and chemical compounds, and provides mechanical resistance. Most of that function is played by the stratum corneum. Characteristics of the barrier: physical barrier through keratinocytes attached together via cell-cell junctions and associated to cytoskeletal proteins, which gives the epidermis its mechanical strength. Chemical barrier through the presence of highly organized lipids, acids, hydrolytic enzymes and antimicrobial peptides. Immunologically active barrier through humoral and cellular constituents of the immune system. Water content of the stratum corneum drops towards the surface, creating hostile conditions for pathogenic microorganism growth. An acidic pH and low amounts of water make it hostile to many microorganic pathogens. The presence of non-pathogenic microorganism on the epidermis surface help defend against pathogenic one by limiting food availability and through chemical secretions. Factors that alter the barrier: psychological stress through an increase in glucocorticoids compromises the stratum corneum and thus the barrier function. Sudden and large shifts in humidity alter stratum corneum hydration in a way that could allow entry of pathogenic microorganisms. Skin hydration. The ability of the skin to hold water is primarily due to the stratum corneum and is critical for maintaining healthy skin. Lipids arranged through a gradient and in an organized manner between the cells of the stratum corneum form a barrier to transepidermal water loss. Skin color. The amount and distribution of melanin pigment in the epidermis is the main reason for variation in skin color in Homo sapiens. Melanin is found in the small melanosomes, particles formed in melanocytes from where they are transferred to the surrounding keratinocytes. The size, number, and arrangement of the melanosomes varies between racial groups, but while the number of melanocytes can vary between different body regions, the numbers remain the same in individual body regions in all human beings. In white and Asian skin, the melanosomes are packed in aggregates, but in black skin they are larger and distributed more evenly. The number of melanosomes in the keratinocytes increases with UV radiation exposure, while their distribution remains largely unaffected. Clinical significance. Laboratory culture of keratinocytes to form a 3D structure recapitulating most of the properties of the epidermis is routinely used as a tool for drug development and testing.